Welcome to Chapter 13. So these are going to be the microbe-human interactions, specifically humans, but this would be all mammals as well. And so this is going to be, again, it's kind of sucks that we're in the middle of a pandemic right now, but it's kind of cool because we get to use that as an everyday example because we're living it right now. And so we're going to be talking about infection, disease, and epidemiology. So if you ever feel alone, you're never truly alone because the human body has a ton of microbes with us at all times. So the human body exists in a state of dynamic equilibrium. Equilibrium is something, homeostasis is something that we have driven into your brain since the beginning of all science classes, okay? So that's going to be finding a happy medium between, not too cold, not too hot, for instance. Our physiologic normal dynamic equilibrium for our temperature is 37 degrees Celsius or 98.2 degrees Fahrenheit, right? Many organisms interact between the human body and the microorganisms involve the development of biofilms. We've talked about that, like the film on your teeth in the morning after you sleep at night is a good example. We also have microbes that colonize the body, and it can involve a constant give and take. So, stomach microbiota, right? If we have too much of a good thing, it can be bad. If we have too much of a bad thing, it can be really bad, right? So it's a constant give and take of trying to find out this equilibrium and stay at that equilibrium level. So when talking about contact, colonization, infection, and disease, microbes that engage in mutual or commensal associations, these are going to be like, okay, they're mutual. It's like a mutual agreement. So these would be like our normal or resident flora. Indigenous flora, those indigenous means natural, right? Or our microbiotes, right? Then we have infections, and this is a condition in which pathogenic microbes can penetrate their host defenses. It can enter the tissues, and then they can multiply and kind of hijack and take over. Then we have a pathogen, which is an infectious agent, COVID. See what I did there? I'm so funny. Oh my God, you guys. All right, and then we have infectious disease, and this is an infection that causes the damage or disruption to tissue and organs. So looking at this with the contact, colonization, infection, and disease, well, this is going to be talking about your infection portals. Okay, so this is figure 13.1 in your book, and this is the associations between microbes and humans. So the effects of contact with microbes can progress in a variety of different directions, ranging from no effect of the colonization and to infection and disease immunity. The example shown in this picture here follows the possible events in the case of contact with a pathogen such as Staphylococcus pneumoniae or the pneumococcus. This bacterium can be harbored harmlessly in the upper respiratory tract, but it also may invade and infect the ear, cranium, and lower respiratory tract. So basically here you would have contact, and it can either colonize or then it can go from a colonization into an invasion where the microbes cross the lines of defense and enter into sterile tissues. Then we have the infection, which is going to be the pathogenic microbes that are going to multiply in the tissue. And then basically a carrier state is going to develop and the microbes are established in tissues, but the disease is not yet apparent. Then down here, after the infections, we can have defenses that hold the pathogen in check, and it'll be yay, and then the immunity or repair of damage occurs. Or we can have the effects of microbes that result in injury or disruption to tissue, and this can cause morbidity or mortality. So morbidity is like a really bad effect, like it's almost deadly. Mortality is obviously when it is deadly. Okay, and so blue here would be the purple, the action of microbes. The nothing highlighted would be, or I guess it's light blue, would be the beneficial effects, and then this pinkish color would be the adverse effects. Okay, so you can see basically it can go either way. If your body's good at fighting things off and it's lucky and it fights it off, then awesome. But other than that, it can actually turn into a potentially mortal morbidity, mortality, a bad disease, and potentially take you out. So starting with the good guys, okay, we have resident flora. These are guys that are going to just hang out on our body at a normal time. So most areas of the body in contact with the outside environment harbor resistant microbes. Internal organs, tissues, and fluids are microbe-free. Okay, so we did this in microbiology lab. I had you touch plates, and to try to grow finger microbes. We had you scratch your scalp over top of the plate, right? And so we know that we do have some normal things. We had to do body swabs, behind your ear or, between your nose or things like that. But if you think about it, our internal organs, we want them to be pretty microbe free, right? And so this is why, like, if you are getting a spinal tap for whatever reason, they make sure that it's clear and not growing microbes because you're not supposed to be growing microbes there. Okay. We have transient microbes and we have residential microbes. So transients are microbes that occupy the body for only a short amount of time. So think of like a transient person. They hop from place to place to place. Where residents, I am a resident of Big Rapids, Michigan, and I'm in this lovely house. And these are microbes that have become established. So looking at figure or table 13.1, these are sites that harbor or host normal resident microbes. So this would be like your skin, your upper respiratory tract, your GI tract, your opening of your urethra, your external genitalia, your vagina, external ear, and your ear canal, and then our external eyes, such as our lids, lashes, and follicles. So it's okay to have normal things growing there. The bacteria flora benefit the host by preventing overgrowth of harmful microbes, and this is called microbial antagonism. We also have endogenous infections, and these occur when the normal flora is introduced to a site that was previously sterile. Okay, and so this is figure 13.2. Okay, the origins of microbiota in newborns. Once a newborn baby emerges, it goes through the second phase of colonization from many different sources. This includes exposure to the birth canal, breast milk or other foods, family members, medical personnel, and the environment. Within a short time, the infant has developed its initial microbiota in major areas of the skin, oral cavity, and intestine. So basically, it comes out of a sterile environment, right? The mom's uterus. And then that's why people are like, before you hold my newborn baby, make sure that you wash your hands. Make sure that you didn't just come from the barn, Janet, stuff like that, right? Because you do want them to be exposed to things, but not in an overwhelming rate. So for instance, again, the uterine contents are normally sterile and they remain so until just before birth. And then breaking that fetal membrane is going to expose the infant, and therefore all subsequent handling and feeding will continue to introduce flora that will become normal for that baby. So with this figure, this is going to be, it says 13.3. And so this is life on humans. So sites containing well-established microbiotes and representative examples of them. So you can read through this and you can see that in our skin, it's normal to have Pseudomonas, Micrococcus, Staphylococcus, Streptococcus bacteria. For fungi, it's normal to have Canidia, and etc. And then for our arthropods, it's normal to have skin mites, which kind of sounds gross, but it's totally true. That's what dander is. So like dust and dander from around the house is you shedding these things, right? You have your GI oral cavity where it's normal to have strepto and lactobacillus, a lot of different things, right? Large intestines, etc. So this is a very good table to summarize the normal guys, right? And then remarks on where they only live. So for instance, on the skin, the microbes live only in the upper dead layers of the epidermis, glands, follicles, the dermis, and layers below are sterile. So it's just those biota live on the surface, but not deep inside. So looking a little bit more at the flora of the normal human skin, the skin is the largest and most accessible organ. There are two cutaneous populations. There are transients, kind of, again, think of a transient, they pick on them, and I don't know, but like hobos or homeless people, those are normally known as transient people. Love them, they're usually very, very, very nice. But our transient microbes are going to be influenced by our hygiene, where our resident microbes, those are the ones that are stable, predictable, and they're less influenced by hygiene. Okay, so this here is figure 13.3, and this is the landscape of the skin. So in A, which is this one here, this is the epidermis, along with the associated glands and follicles that are colored, and they're going to provide rich and diverse habitats. The non-colored regions, such as the dermis and the subcutaneous layers, are free of microbes. And down here in B, you can see a highly magnified view, which is 7,500 times normal viewing of skin layers reveals rod-shaped and coccyx-shaped bacteria adhering to the skin cells, and the skin cells are blue. Okay, so you can see our normal guys. What about talking about our flora of the GI tract? Well, the GI tract is a long, hollow tube that is bounded by mucous membranes, our mouth and our anus, right? The tube is exposed to the environment. When I open my big old mouth and I drink some coffee or, eat food or whatever, we're exposing that to the environment. And the variations in flora distribution due to the shifting conditions such as pH, oxygen, tension, and anatomy. We have our oral cavity, our large intestine, and our rectum that can harbor appreciable or good flora. Okay. Can't pick up coffee without having a sip of it. Okay, so again, this is figure 13.4, and this is showing you the areas of the GI tract that shelter major communities of resident microbes that are highlighted. Okay, and then the non-colored areas, which would basically be the stomach and the small intestine, they do not harbor residents of significant numbers. Okay, so basically right when we enter and right before we exit, that's where we're going to have those flora. What about our mouth? I had you guys do a mouth culture, right? And the flora of the mouth, this is the most diverse and unique flora of your entire body because there are numerous adaptive niches, and their bacteria's count of saliva is 5 times 10 to the 9th power of cells per milliliter. That's a lot. But we need those guys, right? So we actually start our digestive processes in the mouth. We have those lipases, right? Or no, it's proteases that are going to start breaking down your sugars and your proteins and stuff like that. Mostly your sugars in your mouth. What about moving on to our large intestine? Well, this has a complex and profound interactions with the host, okay? We also have a very high microbial load. So it's 10 to the 8th to 10 to the 11th microbes per gram of fecal material or poop, okay? The intestinal environment favors anaerobic bacteria. Ain't no oxygen getting up there, guys. So we're going to have those anaerobic, or the ones that are going to be growing without oxygen. And then the intestinal bacteria contribute to intestinal odor. Have you ever noticed that depending on what you eat, your poop smells different? So this is why, because different ones are going to grow and thrive depending on what microbes or what nutrients they have to grow off of. So looking at the flora of the respiratory tract, well, this is figure 13.5. And so we have our oral streptococci, and this is the first organisms that are going to colonize. It's the first area to make contact, right? So it's going to be the first one to make a colony. You have your nasal entrance, which is in the nasal vestibule, and the anterior nasal pharynx, and that's where Staph aureus is going to tend to grow. You have your mucous membranes of the nasopharynx, and this is where Neisseria are going to grow. Then you have your tonsils and your lower pharynx, and those are where your Haemophilus are going to grow. All right, so again, this is the moist mucus blankus of the nasopharynx. It has well entrenched resident microbes indicated by color, the yellow, and some colonization occurs in the upper trachea, but the lower regions, such as our bronchi, their bronchioles, and our lungs, lack resident microbes. This is a good thing, guys, because we want our lungs and the little alveoli to be able to exchange gas without having to work through crud. Okay. What about flora of the GI? The GI tract, the genitourinary tract. So that's not GI because GI is intestinal. Regardless, the general urinary tract. Okay. Well, these are sites that harbor microflora. In the females, that's our vagina and the outer opening of the urethra. Where in males, that's going to be the anterior urethra. Okay, so you can see in the yellow areas here, basically where microbes can enter and how far they can get. So the changes in physiology are going to influence the composition of the normal flora. For example, in the vagina, when we have differences or cycles of estrogen levels glycogen, sugar levels, and pH, that's going to be affecting what microbes are going to be able to grow down there. And there's a lot of stuff going down in that area all the time. Okay, so our pH changes depending on the time of the cycle we are in, undies, type of undies we wear, type of soaps we use, etc. Okay, so this is figure 13.6, and it's just basically exactly that, the female and male genitourinary residence and the location indicated by color. So what do we do to maintain the normal resident flora? Well, we need these guys. So you can't ever be microbe free. It would kill you. Okay, so we need to have these normal flora, and they're essential for the health of humans. And so the flora are going to create an environment that may prevent infections and can enhance host defenses. So they can help us. Okay, antibiotics, dietary changes, and diseases can also alter those flora. And then we can talk about probiotics, okay? And these are introducing known microbes back into the body. So sometimes, like, if you have tummy problems or you've been on a long scope of antibiotics, it'll kill your normal GI flora. And so what they say is eat yogurt, right? Drink kombucha or, eat fermented foods, because that's going to be putting those good probiotics back into your body. My amazing daughter, she's going vegan. So she has to make sure that she is taking her probiotics and stuff to make sure that, things in her body are staying normal. Okay, so these are all just very important for maintaining and normalizing our resident flora. So what are some major factors in the development of an infection? Well, this is table 13.4. So it's not in my book right now, but basically this is, the table is, sorry, right here it is. These are factors that weaken host defenses and increase susceptibility to infection. Basically, our babies and our older people. So that's why in the middle of this COVID thing, we were originally very concerned about infants and the elderly, because they tend to have weaker immune systems. Therefore, they can become more susceptible to infection. What about genetic defects in immunity and acquired defects in immunity? Okay, think about if you're immunocompromised, you're not going to have that immune system to be fighting off these guys. Or if you have acquired defects in immunity like AIDS, you're not going to be able to fight off of these normal infections. Surgery and organ transplants. Okay, you're opening a sterile environment and potentially giving an introduction of, non-resident flora. Then organ transplants, you have to make sure to, again, you're replacing an organ. Make sure that it's very sterile before you plop it in there, and make sure that you're on antibiotics, antimicrobials for a while afterwards. What about organic diseases such as cancer, liver malfunction, or diabetes? Again, all of these can throw off your normal flora. Okay, and so what you have is the portal of entry where the microbes can invade the barriers. Then they can adhere or attach to the host cells. Then they make it into the pathway of the cells by invading them, and then they grow, they multiply, then they infect the target, human body, and then that can potentially lead to disease. Okay, and then those microbes are going to damage those tissues, and then you can exit sometimes, right? And hopefully we treat it and it doesn't kill us, right? And that's what this means. And so that's when we are going to remove the microbes from the host. So there are major factors that influence the development of an infection. The first one is truth pathogens, and these are capable of causing diseases in healthy people with normal immune defenses. This would be like the influenza virus, the plague bacillus, and the malarial protozoan. We also have opportunistic pathogens, and these cause diseases when the host defenses are compromised or when they grow in the part of the body that's not natural to them. And an example of this would be Pseudomonas or C. albicans, right? Get these in a non-normal area, and it's going to cause some problems. Severity of the disease is going to depend on the virulence, or how aggressive these are of the pathogen. Also, the characteristics or the structure that contribute to the ability of the microbe to cause disease, and that's called the virulence factor. Okay, so basically, how aggressive they are is how virulent they're going to be. So, how do things become established? Well, we have a couple different portals of entry, and these are a characteristic route that a microbe will be able to follow to enter the tissues of the body. So we have exogenous agents, and these originate from a source outside of the body. And then we have endogenous agents, and these are the ones that are already existing on or in the body. And these are our normal flora. But again, they can get out of hand. So this is figure 13.8, and this is showing you the different portals of entry. So infectious microbes first enter the body through the characteristic routes, and most microbes have adapted to a specific means of entry. So one of these, for instance, the uterus, right, is an internal portal of entry as it may allow microbes to access the developing fetus. Okay, so if it gets through the vagina and the cervix, then it can have a direct entry into that growing baby, right? Some other portals of entries are the skin through nicks, abrasions, punctures, incisions. So I have horses, you guys know that, right? And so I'm constantly doing things in the barn or riding or whatever, and I tend to cut myself and whatnot. And so I remember, oh, this was great. I dropped a saw on my hand a couple years ago. I have a nice little scar. You guys probably can't see, but it's right on my knuckle because I literally dropped a saw blade on me because I'm brilliant like that. And so when I went to the barn and when I did things, I made sure to really wrap it up because that was an open port of entry. And I wanted to make sure that I wasn't getting normal, but also abnormal flora in there. So, when I went to the barn, I made sure to really protect it. And then when I left it, I made sure to really clean it. Same with when I broke my finger. At the same time, I happened to slice my finger on metal part of my trailer, my horse trailer. You would think that I should just not ride horses, right? But I'm stupid like that. All right, so anyway, we have different portals of entries. And so with the skin, we have nicks, abrasions, punctures, and incisions. For the GI tract, we have food, drink, and other ingested materials. For our respiratory tract, we have oral, and nasal cavities. And then the urogenital tract, we have sexual interactions and displaced organisms that can get placed from penetration and, the guy's thing is outside and it's exposed to different microbes. And then when it goes into the vagina, right, it's a direct contact into your internal organs, right? Potentially. And then you also can have transplacental, so going through the placenta to the baby. So this is figure 13.6, and this is the incidence of the most commonly STDs from 2008, okay? So you have trichomonas, you have HPV, you have herpes, you have chlamydia, gonorrhea, syphilis, hepatitis, new HIV and infections, and AIDS diagnoses. So you can see this is a little outdated, but it pretty much holds true. So what about pathogens that can infect during pregnancy? Well, this is figure 13.9, and this is detailing the transplacental infection of the fetus. So on the left is going to be the fetus in the womb, and then when you look here, it's going to be a closer view. The microbes are shown penetrating the maternal blood vessels and therefore entering the blood pool of the placenta. And then they subsequently travel into the fetal circulation by way of the umbilical vein. So you can see that they get across and then they can hop the highway and go right into the vein and the artery, which is going to potentially infect the baby. So looking at storch, these are the common ones, right? This would be syphilis, toxoplasmosis, other diseases such as hepatitis B, AIDS and chlamydia, rubella, cytomegalovirus, and herpes simplex virus. These can all be transported to the child through the mother while it is incubating. Okay, so it says not all neonate. My book is on page 412 underneath table 13.6. It says not all neonatal infections occur through the placenta. Some, such as herpes simplex and chlamydia, develop perinatally when the infant is contaminated by the birth canal. The most prominent infections in fetus and neonates are grouped together in a unified cluster known as the acronym STORCH. And that is the medical personnel are on alert to detect. So again, STORCH stands for syphilis, toxoplasmosis, other diseases such as hepatitis B, AIDS and chlamydia, rubella, cytomegalovirus, and herpes simplex virus. The most serious complications of STORCH infections are spontaneous abortion, congenital abnormalities, brain damage, prematurity, and stillbirths. Okay, so just be aware of those. So what is the requirement for an infectious dose? Well, this is the minimum number of microbes that are required for an infection to proceed. So microbes with small IDs have greater virulence. Okay, they have a higher viral load, and then they lack of an ID will not result of an infection. Okay, so this is figure in this book it was 13.7. In the newer version of the book that I have, it's 13.6, so right around there. Okay, and so basically to get measles you only have to have one exposure to the virus, and it's through respiratory tract. For Q fever you only need to be exposed to 1 to 10 cells, respiratory again. So the plague is a little bit higher. Anthrax, you need to have a more exposure to the cells. It's not just one, which is good. Same with typhoid and cholera. Okay, but again, stay away from these things because you don't want to deal with them. So what about attaching to the host? Well, this is called adhesion. When you adhere to something, you attach to something. So this is where the microbes gain a stable foothold in the portal of entry. And this is dependent on the binding between the specific molecules on the host and the pathogen. So examples would be fimbriae, flagella, glycocalyx, the cilia, suckers, hooks, and barbs. So these are different ways that microbes can adhere or attach to the different cell surfaces. So this is going to be figure 13.10. And so in A here, it's showing the different fimbriae, and these are minute bristle-like appendages. In B here, it's adherent extracellular capsules that are made of slime and other sticky substances. And then C here, these are viral envelope spikes. And you can also look further into this by looking at this table, which in my book is 13.7. And so these are the adhesion properties of microbes. So you can see specifically that gonorrhea is going to attach through the fimbriae, attaching to the genital epithelial cells, where AIDS is going to be viral spikes that adhere to the white blood cell receptors, etc. So this is a great summary table. What about surviving these host defenses? Well, an initial response to host defenses come from phagocytosis. We have antiphagocytic factors, and these are used to avoid phagocytosis or that Pac-Man eating, right? Species such as Staphylococcus and Streptococcus, they produce leukocidins. Geez, sorry, my brain's off. Leukocidins, and these are toxic to white blood cells. We also have a slime layer or a capsule that can form, and then therefore, because of this, it's kind of putting this protective barrier around it, and it makes that Pac-Man or the phagocytosis eating difficult. And so the ability to survive the intracellular phagocytosis is dependent on these. And so this is a close-up, this guy right here, of figure 13.11, talking about the pathological effects of virulence factors on host cells. And so for this guy over here, you can see the blocked phagocytic response. So bacteria have a property that enables them to escape phagocytosis, or like I said, that Pac-Man eating, and therefore continue to grow and cause further infections. And so what happens is this capsule is going to encapsulate it and therefore block the Pac-Man from being able to eat it. Okay, so it is a self-preservation type method or mode. What about how we enter the host tissues? Well, some pathogens produce a secretion system to insert specialized virulence proteins directly into the host cell. So again, this is discussed in figure 13.11. And what happens here is that we have adhesion or gluing attaching by the fimbriae. And the fimbriae are those little, the little finger looking protrusion. Then we have a release of proteins that are going to be included in the secretory system. Then this causes a disruption of actin and ruffling. Okay, and then the cell is going to be engulfed or pulled into the vacuole, like vacuum or pull it in. And then the microvilli are going to die. Then the salmonella, in this case, is going to multiply internally, and then it moves out of the cell and it goes into the deeper tissues. And so this is one way that it can invade or enter host tissues. Okay, and so salmonella has a unique system to invade intestinal cells. After adhering to the cell's microvilli with its fimbriae or finger-like projections, it injects proteins into the cell with a probe-like structure. These proteins disrupt the actin filaments and cause the cell structure to form a series of ruffles that therefore surround the bacterium and pull it inside. And this becomes shielded within a vacuole, and therefore it begins to grow. And from here, it can gain entrance into nearby tissues. And so that's how Salmonella is active. What about when we cause a disease? Okay, we have virulence factors, and these are traits that are used to invade and establish themselves in the host. And this also determines the degree of tissue damage that occurs. And this is therefore how severe that disease is going to be. So we have exoenzymes, and these are the ones that are going to dissolve extracellular barriers, and therefore penetrate through or between cells. Okay, and you can see here, okay, exo outside, and it's going to be penetrating through and between. Then we have our toxogenicity, and this is the capacity to produce toxins at the site of multiplication. So that's here. So back to this guy. Again, this is figure 13.11. Bacteria produce extracellular enzymes that dissolve barriers and penetrate through or between cells to invade those underlying tissues. Where here in toxins, the toxins, primarily exotoxins, are secreted by bacteria, and therefore they damage the target cells, which therefore die off and begin to slough off. What about talking about bacterial toxins? This is a potent source of cellular damage, okay? There are two different types of bacterial toxins. We have an endotoxin, and this is where a cell that is not secreted, but it's released after the cell is damaged. And these are composed primarily of lipopolysaccharides, or LPSs. And this is part of the outer membrane in a gram-negative cell wall. We then have those exotoxins. So remember, endo means in, exo means out, okay? And then toxin is going to be toxic, okay? So the exotoxins, these are toxin molecules that are secreted by a living bacterial cell into the infected tissue. So there's a strong specificity for a target cell. They are hemolysins, so they're going to be lysing blood, or hemo, right? Blood is hemo. We also have AB toxins, and A stands for active and B stands for binding. So looking at these guys, this is going to be figure 13.12. Well, let's see if we can get you to see the rest of this here. Sure, there we go. So again, this is figure 13.12, and this is the origins and the effects of circulating endotoxins and exotoxins. So in A here, the exotoxins are given off by live cells, and they have a highly specific target and therefore highly specific physiologic effects. So the target organs are damaged, like the heart, muscle, and blood cells. Intestinal tracts can show dysfunctions from a result of these endotoxin invading. What about B here? This is the endotoxin, and this is given off when the cell wall of a gram-negative bacteria disintegrates, and it has therefore a more generalized physiologic effect. So the general ones, instead of a targeted effect, is going to be fever, lethargy, feeling very just bleh, and then aches and shock. So this is a nice little summary of comparing endotoxins and exotoxins. In my version of the book, it's table 13.8. But basically, it's telling you the exotoxins versus the endotoxins, the toxicity. Exo is toxic even in small amounts, where endotoxins are only toxic in higher amounts. Effects on the body, types of sources, etc. So this is a great little summary table. What about these AB exotoxins? Well, basically, this is figure 13.13. So this is the actions of AB toxin Coromycobacterium diphtheriae. So the B part is going to attach to a specific receptor on the cell membrane, and then the toxin is going to be engulfed into the vacuole. And then the A and B parts are going to separate. The A portion is going to be released into the cytoplasm, where it blocks the synthesis or the making of those proteins that are required for normal cell functions. And then the diphtherial toxins is going to be highly destructive for the cells, especially those of the heart and the nervous system. So A, the active component, is attached to the B binding component, and then the A component inhibits the cellular protein to cause the cellular damage. So what about the process of infection and disease? Well, there are 4 different stages of clinical infections. Okay, and so this is figure 13.14, the stages in the course of infection and disease. So the dashed line represent periods with variable length. Okay, so you can see that here. So the first one is going to be the incubation period, which is this blue guy right here. And this is the time from the initial contact with the infectious agent to the appearance of the first symptoms. The agent is multiplying, but the damage is insufficient, so it's not enough to cause symptoms. And this can last from several hours to several years. So that's very variable, therefore it gives you a dashed line. You have your prodromal stage, and this is vague feelings of discomfort with non-specific complaints, just kind of feel bleh, right? And then you have your period of invasion here, and this is where the microbe multiplies at high levels, it becomes well established, and it is more specific to the signs and symptoms. And then you have your convalescent period down here, and this is where a person begins to respond to the infection and the symptoms decline. So we start to fight it off, right? And therefore it can go away. So this is going to be that process of infection and disease. So what about patterns of infection? Well, we have either localized infections, systemic infections, or focal infections. And so this is going to be table 13.15, and this is the occurrence of infections with regard to the location and sequence. So over here in A and B, in A, the localized infection in which the pathogen is restricted to one specific site. For this one, it happens to be like in the armpit. Where B, it goes systemic. So this systemic infection in which the pathogen spreads through the circulatory system to many different sites. Okay, so localized, it's going to remain confined to a specific tissue. Systemic, it spreads and hops on the bloodstream express and usually goes to other places. Okay, and then this focal infection again is going to be when the infectious agent breaks loose from a local infection and is therefore carried to other areas and other tissues. So C here, a focal infection occurs when initially it was a local infection, but circumstances caused the microbe to be carried to other sites systemically, or using the system as a highway, right? We also have a mixed infection, which is down here, and this is where the same site is infected with several microbes at the same time. And that can lead to E here, which is a primary and secondary infection, where a primary infection is the initial infection, and then the secondary infection is going to be another infection by a different microbe. Okay. So in primary secondary infection, the initial infection is complicated by a second one in the same or a different location and is caused by a microbe. Okay, so for instance, we have them mixed all in one spot or specific but in two different locations. Okay. What about patterns of infection? Well, we have acute versus chronic. So same definitions apply. Acute comes on rapidly with severe but short-lived effects. Where chronic infections, these are going to progress and persist over a long period of time. So this is going to be, in my book, it's figure 13.9. So these are common signs and symptoms of our infectious disease. Our signs could be fever, septicemia, microbes in the tissue, abnormal chest sounds, skin eruptions, etc. And other symptoms could be like chills, pain, nausea, fatigue, chest tightness, itching. So a whole bunch of different signs and symptoms. Okay. So the earliest signs of the disease as a result of the infection of the body defenses, and these usually are going to be fever, pain, soreness, and swelling. So your body's like, yo, something's not right. Let's try to fix this, okay? And then there are signs of inflammation, such as edema or swelling, accumulation of fluid, granulomas and abscesses. And these are where we have walled off collections of inflammatory cells and microbes. And then also lymphadenitis, and this would be like your swollen lymph nodes when you can feel, like, for instance, your neck and you can feel a swollen lymph node. What about signs of infection in the blood? Well, they're changing the number of circulating white cells, and we can have leukocytosis, and this is going to cause an increase in the white blood cells. We have leukopenia, and this is causing a decrease in white blood cells. You have septicemia, and this is where microorganisms are multiplying in the blood, and they are present in very large amounts. Then you have bacteremia, and this is small numbers of bacteria that are present in the blood, and it's not necessarily multiplying, but they are there. Then you have viremia, and this is where there's a small number of viruses. Look for those root words: bacteremia, bacteria; viremia, viral, virus. And so the small number of viruses is present, but again, not necessarily multiplying. What about infections that go unnoticed? Well, these are called asymptomatic or subclinical infections. And although you're infected, the host doesn't necessarily show any signs of the disease. So it's an inapparent infection. So the person doesn't necessarily seek medical attention because they don't know that anything is wrong. Think of COVID carriers in 2020, right? They don't think anything's wrong, so they don't go get treated, and then they're like a super spreader, right? What about portals of exit? Well, pathogens have to, when they come in one way, they have to go out a way too. So pathogens depart by a specific avenue, and this greatly influences the dissemination of the infection. So we have respiratory, so we're using like our lungs and stuff. So your mucus, your sputum, your nasal drainage. So if I sneeze or I cough up a hairball, just kidding, I have cats. Right, this is going to be using our respiratory system to exit. We have skin scales. You can have skin cells slough off. You can have the poops, right? And so you can have a fecal exit. You can have the urogenital tract exit, and you can also have removal of blood. So this is figure 13.16, and this again just shows major portals of the exit from infectious diseases, where pathogens are released from the body through secretion, excretion, discharge, or shedding. All right, what about persistence of microbes in pathologic conditions? Well, apparent recovery of hosts does not always mean the microbe has been completely removed. We have a period of latency. And so this is where after the initial symptoms in a certain chronic disease, the microbe can periodically become active and produce recurrent disease. And the person may or may not shed it during the latent stage. We also have a chronic carrier, and this is a person with latent infection who can shed the infectious agent. You also have sequelae, and these are long-term or permanent changes that can occur to the tissues or the organs. What about sources and transmissions of these microbes? Well, you can have a reservoir, and this would be the primary habitat of the pathogen in the natural world. For example, a human or an animal carrier, soil, water, or plants. And then we have a source, and this is where we have the individual or the object from which an infection is actually acquired. So talking about those living reservoirs here, we have a carrier, an asymptomatic carrier, and a passive carrier. Well, a carrier is an individual who inconspicuously shelters the pathogen and spreads it to others. And they may or may not have experienced the disease due to the microbe. We have an asymptomatic carrier, and these are ones that show absolutely no symptoms. They don't know that they have it, right? And so they're spreading it, not knowing it. Then you have a passive carrier, and this is a contaminated healthcare provider that can potentially pick up a pathogen and transfer them to other patients. Like, wasn't it Typhoid Mary was the original one, right? And so this is figure 13.17. And so this is just showing different pictures, and the other part of it is on the next slide. So this one is C, which is the passive carrier. And so this is a passive carrier, is contaminated but not infected, and can spread the pathogen through physical contact. So this A is an asymptomatic carrier, and that's infected without the symptoms. And then B here is incubation, convalescence, and chronic carriers can transmit the infection either before or after the period of symptoms. So again, this is figure 13.7. So the asymptomatic carrier shows no symptoms. The incubation carriers can spread the infectious agent during the incubation period. You have your convalescent carriers, and these can recuperate or get better without symptoms. Then you also have your chronic carrier, and these are individuals who shelter the infectious agent for a longer amount of time. So what about animals and as reservoirs and sources? So a live animal, other than human, that transmits an infectious agent from one host to another is called a vector. So the majority of vectors are arthropods, such as fleas, mosquitoes, flies, and ticks. But some larger animals can also spread infections, and these would be mammals, birds, and lower vertebrate animals. Biologic vectors, these actively participate in a pathogen's life cycle, And then we also have mechanical vectors, and these are not necessary to the life cycle of an infectious agent, and they're merely transported without becoming infected. Okay, so we have a couple different ways of transporting things. An infection indigenous to animals, but not naturally transmissible to humans, is called zoonosis. And so this would be like, for instance, my horse can get fleas, or my cat can get fleas. Those fleas don't go to humans. Okay, so that would be a zoonotic disease. So it's specific and stays usually within the animal. Humans don't transmit the disease, the zoonotic disease, to another. And at least 150 zoonotic diseases exist worldwide, and these make up 70% of new emerging diseases worldwide. And I believe, isn't that what they think that COVID started with? Is it was in a bat, and it should have been zoonotic, but you know, things have species sometimes that it's become a problem. So, excuse me, it's impossible to eradicate the disease without eradicating the animal reservoir as well. And so this in my book is figure 13.10. And so these are common zoonotic infections. So this would be like rabies and yellow fever, viral fever, Rocky Mountain spotted fever, ringworm, scabies, etc. Okay. So how do we acquire and transmit infectious disease? Well, we have communicable diseases, and this would be where an infected host can transmit the infectious agent to another host and establish infections in that host. And this is like a highly communicable disease would be called contagious. We also have non-communicable infectious diseases that do not arise through transmission from host to host. And these occur primarily when compromised persons are invaded by her own normal microflora. And then the contact with the organism in a natural, non-living reservoir can occur. Okay, so communicable diseases, very contagious. Non-communicable, it usually doesn't arise from transmission from host to host. It's non-transferable. Okay, what about patterns of transmission? Again, guys, we are in the middle of COVID right now, so we know so much about this. So this is figure 13.9, and these they creep me out because, I mean, I like to play stupid sometimes and not realize how far a sneeze or a cough can travel, but you know. So patterns of transmissions, we can have direct contact, and this is where we have a physical contact or fine aerosolized droplets. We have indirect contact, and this is where it passes from infected host to immediate conveyor and then to another host. And the vehicle can be either an inanimate material, food, water, biological products, or fomites. And you also can have airborne, such as droplet nuclei and aerosols. So again, this is figure 13.19, and this is the explosiveness of a disease. So special photography dramatically captures the droplet formation with an unstifled sneeze. So always sneeze into your elbow, guys. Even the merest, the smallest attempt to cover a sneeze with one hand or elbow will reduce this effect considerably. So when droplets dry and remain suspended in air, they become droplet nuclei. Okay, so again, we're dealing with this with COVID right now too. And this is, I'm pro-masker. I'm not telling you what to do, but this is one of the reasons why masks are important. And I know Baker makes us wear masks, so this is why. So what about this? How are communicable and infectious diseases acquired? Well, basically, there are a couple different ways that this can happen. So this is figure 13.18, okay? So here you have communicable infectious diseases. So we can have them transported through contact, which would be kissing and sex. And examples of this would be Epstein-Barr virus and gonorrhea. You can also have respiratory contact or transmission. This would be where we have the droplet spray, and this would be like the colds and chicken pox. So you can see that they're pretty close together. They're talking without masks. Then we have the little droplets. We also have a vertical transmission. So this would be, for instance, from a mother to a child, and this would be how we can transfer AIDS and syphilis. You can also have biologic vectors such as West Nile virus and malaria, and this is where a mosquito or a fly is going to come and transmit it. So these are all direct transmissions. We also have indirect transmission vehicles, and this would be where we have fomites are contaminated inanimate objects, and this is how we can pass on Staphylococcus aureus. You can also have oral fecal contamination that can lead to both of these types of transmissions, such as food, water, biological products such as Salmonella and E. coli. You can also have droplet nuclei, and these are where dry airborne respiratory secretions can transport, for instance, TB and influenza. These are your airborne microbes. And then you can also have aerosols, and these are airborne animal wastes, such as hantavirus. Disturbed soil can transmit coccidioidomycosis. Okay, so there's a lot of different ways for direct transmission or indirect transmission. What about nosocomial infections? Well, these are diseases that are acquired or they developed during a hospital stay. Okay, so this is figure 13.23. So from surgical procedures, equipment, personnel, and exposure to drug-resistant microorganisms. And we have about 2 to 4 million cases a year in the US with approximately 900,000 deaths. So here is a breakdown of a lot of different prevalence of healthcare association infections, or HAIs. So the relative frequency by body site, these are the most common isolates out of the nearly 500 pathogens that are known to exist and cause haze. And so again, we have respiratory tract, urinary tract, surgical incisions. So my book has a slightly different figure, but basically it's going to tell you that, the blood, the urinary, other, etc. So take a look at those and show how the different ones can be transmitted. So how can we prevent these? Well, this is in my book, figure 13.11, and these are the levels of isolation used in clinical settings. So basically, enteric precautions are gown up and glove up, right? Respiratory precautions would be private room with closed door. Drainage and secretion precautions are glove up and gown up. Strict isolation, private room, right? And then reverse isolation, which is also called protective isolation, would be here. And these are the same guidelines for strict isolation as required. And you also have to have the HEPA airflow, right? And this is what they can prevent the spreading of. So you guys are going to get very familiar with this if you go into the medical field. What about universal blood and body fluid precautions? Well, the stringent measures are taken to prevent the spread of nosocomial infections from patient to patient, from patient to worker, and from worker to patient, and these are called universal precautions. These are based on the assumptions that all patient specimens could potentially harbor infectious agents, so they must be treated with the same degree of care. So this leads us to epidemiology. The heck is epidemiology? Well, this is a study of the frequency and the distribution of diseases and health-related factors in human populations. We have surveillance or watching, and so this would be collecting, analyzing, and reporting data on rates of occurrence, mortality, morbidity, and transmission of infections. We also have reportable, and these are where we have to notifiable diseases that have to be reported to authorities, such as like HIV, AIDS, and COVID, right? We have the CDC, which is known for the Centers of Disease Control and Prevention in Atlanta, Georgia, and this is the principal governmental agency that is responsible for keeping track of all these infectious diseases nationwide. When talking about the frequency of cases, we have the prevalence, and this is the total number of existing cases with respect to the entire population, usually represented by a percentage of the population. We have incidence, and this is the measure of the number of new cases over a certain period of time, and this is compared with the general healthy population. We also have mortality rate, and this is the total number of deaths in a population due to a certain disease. And we also have morbidity rate, and this is the number of people that are affected with the certain disease. Okay. So here's an example of some of the epidemiologic data from the CDC. And so there's a lot of different things here. You can see, patterns of infectious diseases in 13.21, but you can see that the infections of the outbreak strain of Salmonella by date and illness onset, you can see that they track it. Different diseases across the United States. We have TB cases by age group, sex, etc. in the United States. And again, this is kind of old data at this point, but still it holds very true. and reported TB cases by race and ethnicity, again, back in 2008. So they track everything. They're being very helpful in this COVID outbreak, by the way. So this is figure 13.21, and these are the patterns of infectious disease occurrence. So we would have an endemic, and this is a disease that exhibits a relatively steady frequency over a long period of time in a particular geographic locale. So you can see this would be like in the coastal states. You have sporadic, and this is when occasional cases are reported at irregular intervals. So you can see that it's spread all over. Then we have epidemic, and this is when the prevalence of a disease is increasing beyond what was expected. Okay, and this is kind of how COVID started. And then it turned into a pandemic, and this is where the epidemic, so the disease, is going to cross all continents. So you can see that it's everywhere, and this is where we are with COVID right now. Okay. And so that would be that. And then this is a fun little thing on making, a connection. And so this is figure 13.13 in your book. And this is Koch's postulates determining the causative agent of the disease. And so basically what they do, and what we're still trying to do with COVID, is find evidence of a particular microbe in every case of the disease, isolate it from the infected subject, and then cultivate it artificially in the laboratory. So this is how we found out that it was COVID-19. Inoculate a susceptible, healthy subject or laboratory isolate to observe the result. So we give it to a mouse or a rat here, and then we can re-isolate the agent from the subject. Then we can use that to develop like vaccines, etc. So again, when the specimen from a patient that's ill with the infection of an unknown etiology is carried through the isolation procedure. And then we've talked about how we can isolate from cultures with streaking, and then we can put it into a media tube to just inoculate and grow that. So that's how we get a pure culture of that suspected agent is made. Then we have a full microscopic and biological characterization of that microbe. And then we put it into a test animal. In this case, it looks like it is a little mouse, okay? And then you observe the animal for the disease characteristics. You take a specimen from it, and then you can isolate a pure culture and identify it and use it for procedures. So the microbe in the initial and second isolations and the disease in the patient are experimental. Animal must match for the postulates or the hypothesis to be satisfied. Okay, so that is the end of this chapter. And again, it's very relevant in today's age because of COVID. All right, we'll see you guys in class. Thanks.